Hello, my name is Mugabi. I welcome you to Biology Lights. And in this session, we are going to talk about applied genetics. A strain of barley A has a high yield of seeds, but a long stem which is subject to lodging, that is, flattening of earlier of the crop. Another strain B has short, sturdy stem, but lower yield. The genotype of variety A is capital H, small s, that is high yield and long stem. And the genotype of B is small h, capital S, low yields and short stem. Part A. Show how a plant breeder would cross these varieties to produce a high-yielding, short-stemmed variety. Part A. The plant breeder would pollinate plant A with pollen from plant B, or vice versa. The gametes would be from A, capital H, capital H, small h, and from B, small h, capital H, capital S. When the gametes combined in the zygote, the seed would all have capital H, capital S, small h, genotype. Since capital H and capital S are the dominant alleles, the plants would all be high-yielding and short-stemmed variety, the F1 variety. Part B. Explain why this variety would not breed true. Part B. When the A capital H small h capital H small h variety produces gametes, these could be capital H, capital S, capital H, small h, capital S, and small s, which when combined in the zygote would produce 4 varieties of offspring, some of which might be short-stemmed or high yield, short stems low yield, long stems low yield, or long stems low yield. In genetic engineering, a dash from one organism is produced into the dash of any related organism. Fill in the missing words from the following words: chromosome, nucleus, gene, protein, genome. In genetical engineering, a gene from one organism is introduced in the genome of any related organism. What name is given to an enzyme which is used to cut a DNA molecule at specific sites? An enzyme which is used to cut DNA at specific sites is called restriction enzyme. What bacterial cell structures are used to carry the gene intended for genetic engineering? The cell structures in bacteria which carry genes intended for genetic engineering are called plasmids. Name 3 useful products that can be obtained by genetic engineering. Useful products of genetic engineering are chemosis, alpha antitrypsin, hepatitis B vaccine, and insulin. Outline the steps involved in using bacteria to produce human insulin. The gene for insulin is cut from an appropriate strand of DNA using restriction enzymes. Plasmids are extracted from bacterial cells and opened up using the same restriction enzymes, and the insulin gene is inserted in the gap. The recombinant plasmids are inserted into bacteria, which then produce insulin. Give 3 examples of genetic engineering that are intended to improve crop plants. Genetic engineering of crop plants can improve resistance to pests, retard ripening, improve uptake of ions from the soil, increase the vitamins content of the crop. DNA can be split into fragments using restriction enzymes. Part A. Outline the technique used to separate these fragments. DNA fragments in solution are placed at one end of a sheet of gel, and an electric current is applied which separates the fragments. DNA can be split into fragments using restriction enzymes. Part B. What property of the DNA fragments allows this separation? Part B. The size of the fragments determines how rapidly they would move through the gel. Small fragments travel faster and further than larger fragments. The illustration shows the separation of DNA fragments produced from blood sample taken at the scene of a crime, plus those of 3 suspects. Rule horizontal lines through the DNA brands from the crime scene to cross the 3 suspects' profiles. Part A. Which suspect is most likely to be guilty? We have the victim, the crime scene DNA, sample of suspects 1, suspect 2, and suspect 3 DNA samples. Part A. Suspect 3 is the most likely to be guilty. Suspect 3 is the most likely to be guilty. Part B. Explain why you think so. Part B. The greatest correspondence in the position of the bands at the crime scene is with those of suspect 3. What is the special characteristic of stem cells? Stem cells can continue to divide throughout their life. What is the special characteristic of embryonic stem cells? The special characteristic of embryonic stem cell is that they can develop into any kind of cell. What are the possible sources of human embryonic stem cells? Embryonic stem cells can be derived from individual cells of an array embryo, from blood cells in the umbilical cord of mammals, and from culture of stem cells. What would be the advantages of using a patient's own stem cells, for example, blood stem cells, to treat his or her illness? The advantage of using stem cells from the patient being treated with that they will not be rejected by the patient's immune system. Hello, my friends. Thank you very much for participating in Biology Lights. I encourage you to subscribe.